Hello, this is Ben Martinez, and today we're going to be talking about operons and gene regulation. So, this is an operon. An operon exists only in prokaryotic cells, and it is used for gene transcription and enzyme transcription inside of the prokaryotic cells. So, an operon is made up of a promoter, which is used to activate the RNA polymerase to then transcribe the genes and the protein to transcribe the genes into mRNA, which will then be transcribed or then be translated into enzymes to be used by the cell. The operator sits in between the promoter and the control genes, acting as a switch for the RNA polymerase to transcribe or to not transcribe, and the repressor prevents the operator from, or is more or less the action of turning on and off the switch. When the repressor is active and attached, it is blocking the RNA polymerase from continuing the transcription and stops the process. However, when the repressor is gone, the RNA polymerase is able to move freely. Control genes dictate what type of operon and what type of operation is being done. First, let's look at a repressible mechanism. A trip operon is used to create tryptophan. The repressor is not bound to the operator, allowing the RNA polymerase to freely create more tryptophan, to freely transcribe for more tryptophan to be created. However, when there's too much tryptophan in the environment, the tryptophan itself acts as a co-repressor, binding to the repressor, changing the repressor shape, allowing the repressor to bind to the operator, then making the operon inactive. The other type of mechanism would be an inducible mechanism characterized by a lac operon. So this is a lac operon. It is used for creating these enzymes to break down lactose when lactose is present in the environment. When lactose is not present in the environment, the repressor is bound to the operator. When lactose always comes with allolactose. So when lactose is present in an environment, allolactose is also present in the environment. And allolactose is used as an inducer for the operon, for this inducible operon. So this inducer would be the allolactose. Allolactose binds to the repressor, changing the shape of the repressor, preventing it from binding to the operator, allowing the operon to activate and code for the genes used to break down lactose. So let's review. A repressible mechanism, such as a trip operon, is constantly coding to create tryptophan using this operon and the RNA polymerase. However, when there's too much tryptophan in the environment, the tryptophan acts as a co-repressor, binding to the repressor, changing the repressor shape and allowing it to bind to the operator, making the operon inactive, preventing the RNA polymerase from transcribing. In an inducible mechanism, such as a lac operon, the repressor is already bound to the operator until the specific compound is present in the environment, such as lactose, specifically lactose for this operon. When lactose is not present in the environment, the repressor remains bound to the operator, preventing these enzymes that break down lactose from being produced. However, when lactose is present, allolactose is also present, making the inducer present. Allolactose serves as the inducer, binding to the repressor, then changing the shape of the repressor, preventing the repressor from binding to the operator, and allowing the RNA polymerase to code for these enzymes needed to break down lactose. Thank you very much.